AR-15512. Results of Phase 3 studies for Acoltremon ophthalmic solution 0.003%, a novel TRPM8 agonist. TRPM8 are corneal receptors that detect temperature and osmolarity changes on the ocular surface and activate the trigeminal nerve to produce basal tears. Acoltremon ophthalmic solution 0.003% is a potent and selective TRPM8 agonist being investigated for its impact on tear production and ocular surface symptoms. Previously, exploratory results from a Phase 2b study, COMET-1, suggested that twice-daily administration of Acoltremon might increase unanesthetized Schirmer and reduce Sandy symptom scores. Two Phase 3 studies, COMET-2 and COMET-3, were conducted to evaluate the safety and efficacy of Acoltremon in subjects with clinical signs and symptoms of dry eye disease. Eligible study participants had moderate to severe signs and symptoms of dry eye and were not allowed to continue or initiate artificial tears or other treatments that might confound study results. Requirements, study designs, and endpoints were identical for both studies. After participating in a 14-day run-in period with vehicle, subjects were requalified and randomized 1-to-1 to Acoltremon or its vehicle, applied twice daily to both eyes for 90 days. The primary endpoint was the proportion of subjects who achieved 10 millimeters or greater improvement in unanesthetized Schirmer score on day 14. In both studies, Acoltremon treatment had a significantly greater proportion of subjects who met the target compared to vehicle. The pooled analysis produced similar results, finding that, on average, 36.5% more subjects achieved 10 millimeters or greater increase in Schirmer scores compared to vehicle. Over the 90-day study period, the proportion of subjects who achieved 10 millimeters or greater increase in Schirmer score was significantly greater than vehicle at all time points, of which day 1 and day 90 were secondary endpoints. Change from baseline in Sandy score, the key secondary endpoint, was significantly greater with Acoltremon in COMET-2 and numerically greater with Acoltremon in COMET-3 on day 28. Those differences were statistically significant on days 14 and 28 in the pooled analysis. With Acoltremon, reductions in exploratory total corneal staining were observed across the 90-day treatment period. Similarly, reductions in exploratory total conjunctival staining were observed with Acoltremon across the 90-day treatment period in both studies. No serious treatment-related ocular adverse events were reported. The most commonly reported adverse event was instillation site burning or stinging, which occurred in approximately 51% of subjects and were reported as mild in 98% of those subjects. In summary, Acoltremon is a TRPM8 agonist that was evaluated for impact on Schirmer scores and ocular dryness symptoms in two Phase 3 studies. Twice-daily administration of Acoltremon ophthalmic solution, 0.003%, resulted in significantly higher proportion of subjects who achieved 10 millimeters or greater increase in Schirmer score, reductions in Sandy symptom score in favor of Acoltremon, and decreases in total corneal and total conjunctival staining scores. Mild instillation site burning or stinging was the most common reported adverse event.